Today we are going to start new chapter and new unit. New unit is genetics and evolution. In this unit we have three chapters: principles of inheritance and variation, molecular basis of inheritance, and evolution. So we are starting today principles of inheritance and variation. This topic is under genetics. When we think of genetics, we think of Mendel. Mendel is the father of genetics. Mendel was born in the year 1822 on 22nd of July. Mendel was not a biologist, rather he was a mathematician, and he was appointed as a priest in the church. Mendel worked on pea plants for 6 years. He died in the year 1884, and till his death his work was not recognized. 16 years after his death, the work of Mendel was rediscovered by three scientists: Hugo de Vries, Correns, and Schermat in 1900. So once again, remember this, that Mendel work was rediscovered by Hugo de Vries, Correns, and Schermat in 1900. There were some reasons for non-recognition of Mendel's work. At that time, the print media or the media of publications, they were not very strong. Nowadays, we are having local journals, international journals, national journals, but at that time, our publication was weak. So his work was not published properly. And when it was not published properly, it was not globalized. Secondly, as I told, that Mendel was not a biologist, he was a mathematician. So biologists could not comprehend that a mathematician can give some rules or laws for biology. And thirdly, Mendel told that there are certain things known as factors which are inherited from one generation to another generation. Now that is a discrete unit, as at that time we were not having a developed microscope, or we were not having chemical analysis of these factors. So it was just a imaginary, and the people could not think the imaginary thing that could be in the bodies and that can be transmitted from one generation to another. So these are certain reasons for non-recognition of Mendel's work. But now the Mendel's work is the basis of genetics. What are the reasons of Mendel's success? That also we will study now. That Mendel selected all the contrasting characters. He worked on the contrasting characters. Now what does contrasting characters means? Means there is no ambiguity. If we are telling tall, taller, tallest, there may be ambiguity and perception differs from individual to individual. But if I am telling tall and short, that means tall is tall for everyone, short is short for everyone. So these are contrasting characters. And when we are choosing a contrasting characters, there is no ambiguity. Another success was because the large sample size he chose to collect the data. We know that for a reliable data and to reach a conclusion, a valid data is that it has been selected from a very, very large population having a large sample size. So the size or the sample size of the Mendel was very large. And since he was a mathematician, he could analyze that data properly. So data analysis was proper was the third reason behind his success. And by the God's grace or by chance or by his luck, we can say that there were no linked genes in his experiment. Whatever experiment he did, the linkage of the genes was not there. So there are now reasons of selection of pea plants. We studied about the Mendel, about the non-recognition of the Mendel's work, and also the reasons for the success. Now, what are the reasons for selection of the pea plant? Mendel selected pea plants because the life cycle was short. Means within 3 to 4 weeks, we could get the result. Also, pea plant could be self as well as cross-pollinated or fertilized. There were possibilities for self-pollination as well as for cross-pollination. So selfing and crossing became easier. Pea plant shows all the contrasting characters. That we will discuss in the later videos, that what are the contrasting characters. And one more important thing is that seeds do not show dormancy. We know that dormancy of the seed is non-germination of the seeds even if the proper or favorable conditions are applied. Means the sleeping embryo is in the sleeping state. So pea seeds did not show dormancy. And so the time that took for the generation of the new generation was less. And at a time, more number of seeds were produced. So the sample size for every generation was quite large. So this is about what the Mendel did, why he was not recognized, why he has been so successful, and why did he select the pea plant for his experiments. In the next video, I will be discussing about important terms which are used frequently in genetics.